With new imaging and recording technologies like MRI, scalp electrodes, internal brain electrodes, and even genetically engineered light-emitting neurons, why are the basics of brain circuits involved in recognition still a mystery? In this channel, I describe both the beauty and the difficulties studying the brain from a unique and multidisciplinary perspective. The various brain imaging electrical signal recording techniques, both inside and outside of the brain, enable doctors to verify and treat strokes, tumors, locations of intractable epilepsy. All of this has revolutionized medicine and neurology, giving a much better understanding what is going on in diseases and what part of the brain is being affected. In addition, methods like functional magnetic resonance imaging allow to see what parts of the brain are active in live humans. In addition, new imaging technologies is advancing non-human research based on recording neuron activations through marker molecules that change their light properties with voltage. Such markers can be added to the fluid that bathes neurons so that certain ions, electrical salts such as calcium, are detected flowing through neurons. Markers can also be introduced through genes, what's called optogenetics, with virus infections or introduced into the organism's genome from birth. Moreover, not only can neurons be engineered so their voltage will be indicated by light, they can also be engineered where shining a light will activate a neuron, which gives very specific locations to activate neurons and see what they do. So there's huge advances in technology, imaging, genetic control, electronics, and computational power. So why are we still unsure of the basic circuits of the brain? For example, the circuits that function during recognition. It is very difficult to study the brain, even with our technology, because the information that we want to know is laid out in circuits that are both too dense and too spread out. And no one explains this better than the folks at the World Science Festival in their video on connectomes. If the outside of the brain is equivalent to a map of the whole Earth, a neuron would be an individual person, and the most possibly detailed area seen by an fMRI would be a small city in this resolution.
A city about the size of, say, Sheboygan, Wisconsin.
That little blur, that little blob there might be Sheboygan, or we're not sure.
That little blob is a 1 millimeter cube of brain tissue, and it contains a staggering number of neurons.
50,000 nerve cells and about 1 billion synapses.
So right now, there's no way to model an entire human brain by diagramming its micro-wiring. Scientifically for now, we're lost in the foggy limits of brain science.
You would need more digital information than is the actual digital content of the world. What we're working on now, which is to use electrons, which have a much shorter wavelength and give us the ability to resolve every one of these wires. And the way we do this is we take a block of brain and we fill it with a metal called osmium, and we take that block of brain filled with osmium, that little black spot in there, and then we put it in a plastic, very hard plastic, and we put it on a chuck, and we move it up and down against a diamond knife. That's what's going on in that second picture. And then from there, out comes one section after another. They're about 1,000th as thick as a human hair. So we get 1,000 per hair thickness. And then we pick them up on a tape, and then we take a picture of each and every one of them with a very high-resolution microscope. And the pictures are 250,000 pixels by 250,000 pixels, each image, and then we have to take 33,333 sections like that to do a cubic millimeter. It's basically a film strip, and you take those pictures and you stack them up, and you get a stack of images that are now a three-dimensional version of the brain, and then you color in the objects using deep convolutional neural nets to generate the structures in the brain. Here is a dendrite of a nerve cell and an axon talking to that cell in green. And that's one little piece of an area that we did that is just so discouraging. It's so depressing. There's just so much stuff in these brains. There's 1,500 nerve cells. This is 3 billionths of a mouse brain. 3 billion. It took us 5 years to do 3 billionths of a mouse brain when we started. It's just extraordinary. And a synapse every cubic micron, so there are 1,500 synapses in there.
In animals like invertebrates, certain neurons may be huge and can be seen with the naked eye. One can record from them, but even there the system is not well understood. The best understood circuits are the ones for sensing very simple noxious stimuli, or motor neurons governing simple behaviors like a withdrawal reflex or chewing. But understanding circuits of sensory recognition remains difficult because of the density. Electrodes placed on the head may have even worse resolution than the fMRI, but they do have the advantage that they are more sensitive in time. The fMRI records relatively slow changes in neuron state because it records changes in oxygen consumption, not electrical signals directly. Electrodes invasively placed in the head, like ECOG, as is done for epilepsy patients, can give better spatial resolution, but it's still not enough. It is also possible to poke into and record from inside neurons, but even then it's difficult to understand how that neuron affects and is affecting the thousands of other neurons that it connects with. And neurons are rarely recorded in isolation, even if they are part of brain slices. They still have thousands of connections, and in a slice, important circuits may be damaged. Even neurons that are grown in a petri dish survive best when they form networks of thousands of neurons. Now, electrophysiologists have an ingenious way of fixing neuron voltages or currents, forcing the neuron state and revealing effects of individual channels. But such a clamped state is not necessarily a natural state of the neuron within its network in the brain. A neuron may connect to hundreds or thousands of other neurons. Those likely have mechanisms to feedback and regulate the original neurons. In fact, there's abundance of evidence of regulatory feedback or homeostatic adaptation currents, for example, and those are not well understood. And electrical clamping could obscure some of these and other circuit effects. In addition, clamping a postsynaptic cell while getting it to fire with a presynaptic cell may force the rest of the circuit to a state it is never found in the brain. The point is, electrophysiological recordings of neurons are some of the best tools that we have that can both electrically and chemically study neurons and synapses. But because of the same density problems, it still remains challenging to understand the functions of circuits. And even if you know all the neurons, for example, in the worm C. elegans, it has 302 neurons, and all their connections have been charted. But even there are problems understanding the computational function of basic circuits, especially sensory recognition, because of the high degree of feedback, interconnectivity, and density. It is essential to understand the role of the circuit relative to the role of the neuron. So what is the solution? No one method is giving the answer. There needs to be collaboration of as much diverse studies of the brain as possible. Information needs to be taken from multiple clues and find the circuit model that shows most of the phenomena from many aspects: imaging, cognitive studies, reaction times, neuron studies, electrical studies. And it's important that information and the models that are built to capture all of that have the least amount of hand waving. For example, they avoid reconfiguring the model between phenomena. So if you have a contending circuit, you don't want to have that circuit reconfigured for experiment A and for experiment B, for imaging A, imaging B, and then for the cognitive experiments and so on. You don't want models where you have a lot of weights that you have to retrain for each task. You want to find the models with the least amount of retraining or parameters that captures the most amount of phenomena. And I will talk about this more in future videos. If you like this content, please subscribe. I will talk about the different aspects of research and how to combine them all together. And let me know in the comments if there are any topics you would like me to cover. For those that would like to further support, peek into the next videos, books, software, and academic talks in development, please join my Patreon.